Welcome to Week 6 of BIOT 643. This week, we'll be discussing protein technologies. This will be part 1 of this series. During the course of this lecture, we'll be discussing the detection of proteins, using staining, using SDS polyacrylamide electrophoresis, Western blotting, and fusion proteins. We'll also cover the determination of protein structure and the determination of protein interactions. Within this topic, we'll be looking at protein-protein interactions as well as protein-DNA interactions. So first, the detection of proteins. Staining. In terms of staining of proteins, there are numerous available stains. These are often technically challenging and tend to be poorly reproducible. Here we show examples of the two most commonly used stains for protein gels, Kamasi blue and silver stain. Kamasi blue is the most common and has been around for many years. The Kamasi blue requires staining of the entire gel and as a result must be destained in order to visualize the protein. It has relatively low sensitivity and you're only able to detect protein bands which contain around 100 nanograms per band. There are new methods of Kamasi blue staining such as the colloidal Kamasi blue. This is far more sensitive and can detect protein bands in the range of 10 nanograms. Since this stain stains only the protein, no destaining of the gel is needed. Silver stain is a more recent addition and it is more commonly used simply because it has an extremely high sensitivity. In the case of silver staining, this utilizes protein binding of silver ions, which is then reduced into silver metal, creating a visible image. It is highly sensitive in the range of 1 nanogram of protein per band or less. The original silver stain method was not compatible with downstream methods, such as mass spectrometry. However, newer versions of silver staining are compatible with these downstream methods. There are some newer methods that are fluorescent based, and here we compare 4 different methods: the Kamasi blue, the silver staining, and the Cipro orange and Cipro red. The Cipro stains are fluorescent based. Again, these are the same proteins run on the same gels stained with 4 different methods, and you can see that the Kamasi blue has by far the lowest sensitivity. We see an improvement with the silver staining, but clearly the fluorescent staining of proteins gives you a far greater sensitivity than these older methods, and again these are compatible with downstream methods. So let's talk about SDS polyacrylamide electrophoresis. This is denaturing electrophoresis. Electrophoresis separates biological micromolecules according to size. However, a protein's structural size is not necessarily tightly correlated with its amino acid length. And that's because it's also defined by its tertiary structure and its quaternary structure, i.e. whether it forms alpha helices or beta pleated sheets, and its tertiary structure, how you have multiple proteins combining together and how they fold and they form globular structures. A denaturing gel causes a protein's higher order of structure to become undone. This leaves only the primary structure, i.e. the amino acid chain. Electrophoresis can then separate various different protein chains based solely on their length. Molecules called detergents can cause denaturation of the protein structure. One of the most commonly used detergents in molecular biology is sodium dodecyl sulfate, or SDS. SDS is added to a polyacrylamide solution, which is then solidified into a gel. The SDS denatures the proteins and allows them to separate based on their primary structure. It's important to note that the SDS is negatively charged, so the SDS in the solution will bind to the positive amino acids on the protein, and between the SDS which binds to the positive amino acids and those amino acids which are negative, your protein then has a net negative charge. So if you notice in figure A, the sample is added to the top of the gel where the negative electrode is. When the electricity is applied to this, the proteins move from the negative electrode to the positive. And this is because your protein now has a net negative charge, so it moves toward the positive electrode. So as you can see in B, you have a mixture of macromolecules, an electric field is applied, and the proteins move through the gel because of this electric charge. But because of the porous gel, it's separated based on its primary structure, which correlates to its size. So what you see are the smallest molecules move the furthest in the gel, so they're at the bottom of the gel. The medium-sized proteins are in the middle of the gel, and the large proteins remain more at the top of the gel. And this allows you to then calculate the molecular weights based on the migration of a protein through the gel. Another form of electrophoresis is isoelectric focusing. Proteins have a net charge based on the sum of the charges of all their amino acid residues. The protein's isoelectric point, or pI, is the pH at which all net charge equals 0. The isoelectric focusing is a process of separating proteins based on their pI. So in A, you see the low pH on the left and high pH is on the right. So a protein will move through this gel based on the pH of the gel until it reaches the pH at which its net charge equals 0. At that point, the migration stops. So what you end up with is these proteins move, as you see in A, toward the pH that corresponds or closer to their pI or their isoelectric point. And B, once they find this point where their net charge is now 0, since their charge is 0, they cannot move either toward the positive or toward the negative electrodes, so they become stationary. And this then allows you to determine what the protein's isoelectric point is. The next technique we'll talk about is two-dimensional gel electrophoresis. Two-dimensional gel electrophoresis combines both the isoelectric focusing, which we just talked about, and the first technique we talked about, SDS page. And this essentially separates a protein based on both its pI and its molecular mass. In the first dimension, you load the protein sample onto an isoelectric focusing gel. Electrophoresis separates proteins according to their isoelectric point, where their net charge becomes 0 compared to the pH of the gel. So here you add your protein to the top of the gel. An electric field is applied. The proteins migrate through the gel until they reach a pH of the gel where their charge is now 0. And at that point, they stop migrating. In the second dimension, this isoelectric tube gel is then rotated 90 degrees and placed onto the top of an SDS polyacrylamide gel. Electrophoresis now separates the proteins according to their molecular mass or molecular weight in kilodaltons. So as you can see, it's done exactly the same way as normal electrophoresis, but now you have your tube gel is laying on top of this polyacrylamide gel, which is now a slab gel. Electric field is applied, and the proteins are now separated based on their molecular weight. So what you see on the far right are the stained gel showing the proteins as a series of spots separated by their isoelectric point and their molecular mass. And this two-dimensional gel electrophoresis separation of human platelet proteins are shown here on this gel. The spots are proteins that are separated by the isoelectric point on the x-axis, as well as the molecular mass shown on the y-axis. This allows a separation of hundreds of proteins within a single gel. Even proteins with similar molecular weights can be separated from each other based on both their isoelectric point as well as their molecular weight. Next, we'll talk about Western blotting. Western blotting is a technique combining gel electrophoresis, antibody binding, and chromogenic or enzymatic detection. It is also called immunoblotting. After electrophoresis, and this can be SDS page or two-dimensional electrophoresis, the separated proteins are transferred from the gel onto a membrane. This is generally a nitrocellulose membrane, but it can also be a PVDF membrane. Antibodies specific to the protein attach to the protein in the gel. A secondary antibody attaches to the first antibody. This secondary antibody has bound to it an enzyme which catalyzes a detectable enzymatic reaction of a chromogenic substrate. Here we show you a diagrammatic example of Western blotting. You separate your protein sample by SDS polyacrylamide electrophoresis. This separated group of proteins is then transferred to a nitrocellulose membrane. While not shown here, it's important to note that after this transfer, this nitrocellulose membrane is incubated with a protein solution. This generally can be a milk solution or a solution of casein or a solution of albumin, but it's a nonspecific protein because the nitrocellulose membrane can bind any protein which comes in contact with the membrane. So you bind any sites which do not have your specific protein you block with a nonspecific protein solution. Then, where it shows the antibody detection, you incubate with your primary antibody. This is the antibody directed against the protein of interest. Say, for example, you are looking for protein X. Now you incubate initially with an antibody against protein X. After you incubate this with the membrane, you then wash the membrane to remove any excess antibody that didn't bind. If this is a mouse monoclonal antibody, you now incubate it with a second antibody or a secondary antibody. And this would be perhaps if the first antibody is a mouse monoclonal, your secondary antibody would be an anti-mouse antibody. And this is then labeled with, the antibody is labeled with a chromogen. After you incubate this chromogen labeled secondary antibody, you then wash any excess antibody that did not bind away. You now detect the chromogen. This can be an enzymatic, so that you're looking for a colored reaction, or it can be something, a luminescent reagent, so that you're looking for a light reaction. But you react the substrate for which the secondary antibody has a linked enzyme, and this allows you to detect and quantify the protein that was present in the original protein solution. The last group of detection of proteins are fusion proteins. The DNA sequence of a second gene or other useful marker can be added to the DNA sequence of the target gene. For example, you can add a second gene for a green fluorescent protein such as GFP, or an enzyme such as beta-galactosidase, or what's called a histag or a polyhistidine. Upon translation, the two sequences will be translated as a single protein. So you'll have your A, protein of interest, with B attached to it a second protein or peptide which can serve as a marker of that target gene. Here, the protein sequence of the second gene or other useful marker is added to the DNA sequence of the target gene. So here you have your protein of interest with this red tag attached to the end. Such as a GFP, which is a green fluorescent protein. So when this is translated, what you end up with is your protein of interest with the GFP as a single molecule. So next we'll talk about the determination of protein structures. The information in nucleic acids is encoded directly in the sequence of the nucleotides. While the same is true of a protein's primary structure, that is, the amino acid sequence, the activity of a protein is more a factor of its folded conformation, that is, its tertiary structure, and binding with other folded proteins, its quaternary structure. Determination of a protein's three-dimensional structure is key to unlocking an understanding of its activity and function. So one of the methods used for analyzing and determining a protein structure is X-ray crystallography. A purified protein is formed into a crystal. The individual protein molecules form a repeating structure lattice. X-rays are shined through the protein crystal, which diffracts the beam. The diffracted beams are registered by a detector. Finally, a computer analyzes the diffraction and models the protein structure. And here on the right, we show a diagram which gives the basics of this structure. The proteins are formed into the crystal, where the proteins form a repeating structure lattice. X-rays are shown through this protein crystal, and the X-ray beams are diffracted. The diffracted beams are registered by the detector, and here you show this electron-dense map is constructed, and a computer forms and models what the protein structure was in the original protein. Again, it shows the X-ray source is beamed through the protein crystal, where you have diffracted beams against a detector. This gives you a diffraction image. This X-ray diffraction pattern, this particularly shows a photograph of the crystal of myoglobin. The structure is then analyzed by a computer, which generates a tertiary structure of the protein. Again, this is the myoglobin. And here it shows the location of a bound oxygen atom is shown in red. There's also neutron diffraction, similar to X-ray crystallography. In this case, it analyzes the diffraction of a neutron beam. This provides a higher resolution of the atomic structure. And on the right shows an example of this kind of analysis. Another method is nuclear magnetic resonance, or NMR. Nuclear magnetic resonance spectrometry allows the determination of a protein's structure in solution. It does not require the crystallization of a protein, which is often difficult to achieve. So this is a very important distinction from what we just talked about with the X-ray crystallography or the neutron beam crystallography, as this can determine the protein structure in a solution. Nuclear magnetic resonance can show which protons, such as hydrogen atoms within a molecule, resonate as a result of the proximity with each other. And again, the diagrams on the right show how the tertiary protein structure looks, and the resulting NMR image showing, in this case, the number, how the hydrogens at 2 and 5 resonate together, and you can see in this image. Based on the level of resonance detected between two different proton pairs, the distance between the pairs can be calculated. These calculations enable the three-dimensional structure of the final protein to be modeled. Here, we show examples of the structure calculated on the basis of NMR constraints. In A, the NOESY observations show that the protons connected by the dotted red lines are close to one another in space. And B, a three-dimensional structure calculated with these proton pairs constrained to be close together. Some of the amino acid residues can be in varying positions, here we shown in red circles, in different runs. These represent highly disordered parts of the molecule. The protein backbone will often be very constant, enabling the protein's general structure to be determined. In this particular diagram, we show a family of structures, a set of 25 structures for a 28 amino acid domain from a zinc finger DNA binding protein. The red line traces the average course of the protein backbone. Each of these structures is consistent with hundreds of constraints derived from the NMR experiments. The differences between the individual structures are due to a combinations of imperfections in the experimental data and the dynamic nature of proteins in solution. Now we'll turn our attention to protein-protein interactions. All major processes in biology, such as DNA replication, transcription, translation, splicing, secretion, metabolism, cell cycle control, signal transduction, all involve protein-protein interactions. And some of the common methods used to study these protein-protein interactions include the yeast 2 hybrid system, co-immunoprecipitation, fluorescence resonance energy transfer or FRIT, far western analysis, protein arrays, and phage display. When one looks at co-immunoprecipitation, this is used to determine if two proteins are associated in vivo. An antibody binds to a known protein in a biologic solution. The antibody is collected, and unknown proteins bound to the known protein are also collected. So in this case, we have in the diagram a protein lysate or a protein mixture. We have our coupled antibody and our antigen, as well as proteins which interact with our antigen. So in several places you see our sort of red triangle coupled to a blue sort of cylindrical protein. And wherever these proteins interact, when our coupled antibody is added to this mixture, it binds specifically to the antigen. And not only does it bring down the specific antigen, it also will precipitate any protein that's associated with that antigen. And in this case, when we say a coupled antibody, this can be a magnetic bead or an agarose molecule or a Cephadx molecule, but it's some sort of molecule which allows the antibody to be easily pulled down. So this coupled antibody is then spun and washed. We then can elute the proteins from this antibody, and this is usually achieved using an acidic pH, say a pH of 3. A typical buffer would be something like a glycine HCl buffer with a pH 3 or less. Because the antibody binds to the antigen by hydrostatic interactions, this change in pH causes the proteins to dissociate from the antibodies. You now have your antigen and any protein that was associated with that antigen are now released from the antibody, and you can now analyze them. The next method we'll talk about is fluorescence resonant energy transfer. This is based on the transfer of energy from a donor to an acceptor molecule. The transfer and the resulting fluorescence is used to calculate the distance between the two molecules, which is a measure of the interactions between the two. Here we show protein or molecule A and B each have a fluorescence, and the fluorescence resonant transfer or FRET that is shown by this arrow between the two allows you to calculate the distance between the two molecules. The next group of proteins interactions will be discussed are protein DNA interactions. Chromatin immunoprecipitation, or CHIP or CHIP. This is similar to co-immunoprecipitation. However, rather than a protein bound to a protein, this chip captures protein bound to DNA, and it is used to identify the interactions of proteins with DNA, forming a protein DNA complex, such as a transcription factors on promoters. This case, we show a formaldehyde cross-linking in living cells. Then there is cell lysis and sonication in detergent. This breaks up those complexes. You now have the immunoprecipitation with magnetic beads coated with your specific antibody. Here we have, whereas in CHIP, we have reverse crosslink and isolate the DNA. You isolate the immunoprecipitated DNA and you detect it by PCR using primers to specific and control sites. You then quantitate the ratio of specific and control sites in the immunoprecipitated and input DNA. These are the references that may be helpful in reviewing this material.